Hi guys, this is Alicia with Good Morning Sunshine. Today we're on week 3 of vet school. I think we're on day 14 today. Today I had Neurobiology 4 and 5, and then so I had 2 lectures this morning, and then I also had cardiac today, so our first lecture on the heart. And then I had 2 labs. I had a neurobio lab and also a wet lab to do with the thoracic wall. So we gotta take the ribs out of our dog today. Well, I worked on the cat, so I took ribs out of a cat. And we gotta look at the lungs and the pleura of the thoracic wall and the heart. But yeah, so that was pretty cool. So today, again, we started with neurobio, so I'm going to get into that, starting things off. We did support systems of the meninges, the ventricles, the cerebrospinal fluid, which is the CSF. We did blood supply and the blood-brain barrier, which is BBB. So if you hear those, that's what I'm meaning by it. So we have objectives, them being the protection of the brain and spinal cord from physical injury by the meninges and the cerebrospinal fluid, the CSF origins and destinations, the CSF's role in brain tissue and waste disposal, major blood vessels supplying the brain and spinal cord with blood, so nutrients and oxygen, some mechanisms maintaining consistency of the blood-brain supply, and also, lastly, the protection of brain from harmful disruptive chemicals by the blood-brain barrier. So let's start and get into it. See if I can get this to work here. Okay. So we started with how is the brain protected from physical injury? So the brain has several layers of physical protection. It's encased in the skull, but it also has 3 protective membranes covering it. The outermost being the dura mater, and then we have the middle layer being the arachnoid layer, and then we have the subarachnoid space, which includes the CSF. And then lastly, we have the pia mater, which is the most delicate layer. I've talked about a few of these in my past videos, but we're going over them again. These layers are also known as meninges. So basically, that is how the brain is protected from physical injury, with all of these layers, including the skull and the periosteum and the scalp. So in order, it goes from the pia mater, the arachnoid, the dura mater, the skull, and then the periosteum, and the scalp. And the arachnoid, the subarachnoid space is there for cushioning as well. So here I could show you actually a picture. So it has all of these layers that I was talking about before it actually reaches the brain. The spinal cord is also encased in a bony outer region, being the spinal column, and it's also covered by the meninges, the dura mater, the arachnoid, the subarachnoid space, and the pia mater. The meninges run laterally out to the spinal nerves, and these spread out in order to become the epineurium nerves. So where does the cerebrospinal fluid exactly come from? Well, it's produced in cavities, which are ventricles, interconnected cavities in the brain where the CSF is made and flows through. The CSF flows into 2 lateral ventricles that are in the telencephalon, and then it goes to the 3rd ventricle in the diencephalon, and then it flows into the 4th ventricle located in the pons and the medulla, and then it flows out of the brain into lateral, or from the fourth ventricle into lateral apertures to the subarachnoid space, and then surrounds the entire central nervous system with the pia covered. So this is basically all this shows. Shows where the CSF is made here, which I said, and flows through the ventricles of the brain in a series of interconnected cavities. And then over here, basically this is showing you how the CSF flows and how it goes into different ventricles, and then finally covering the entire central nervous system. So what produces cerebrospinal fluid? Well, it is produced by a material called the choroid plexus, which is in each of the ventricles. There are blood vessels poking their way into the ventricular wall. The choroid is a complex of brain capillaries and epithelial cells. The epithelial cells are controlling the ions and molecules from blood vessels into the brain, and what gets into the cavity of the ventricles, thus regulating the composition of the cerebral spinal fluid. So again, CSF produced by the choroid plexus in each of the ventricles. And if it wasn't clear, the choroid plexus is capillary and epithelial cell complex. So where does the CSF eventually end up? Well, it ends up either draining into the lymphatic vessels, which is the more favored process, and those are near the brain, and they permit lymph node immune surveillance of the cerebrospinal fluid, or they can drain into the dural venous sinuses, which are canals that are running in the dura that carry venous blood. These sinuses will drain away out of the skull. The dorsal sagittal sinus is long and wide and runs down the midline of the dural venous sinus. So recall that after leaving ventricles through the lateral apertures, the cerebrospinal fluid, the CSF, in the subarachnoid space bathes outside the surface of the brain and the spinal cord. So then again, it drains into the lymphatic vessels, which are near the brain, or into the dural venous sinuses, which are large venous canals within the dura, which join the venous flow out of the head. So then we got into more specifics of the lymphatic cerebrospinal fluid drainage. So there are lymphatic vessels, which are also called meningeal lymphatics, that lie within the dura itself. The cerebrospinal fluid can gain access to these and drain into them. There are also nasal lymphatic vessels that sit by the olfactory bulb, which also sits near the skull by the cribriform plate, which I've talked about in other sitcoms. There are lymphatic vessels that sit near this plate, near the olfactory bulb, like I said, and the CSF can get access to these lymphatic vessels as well. The lymphatic drainage of the cerebrospinal fluid can result in lymph node immune surveillance of the CSF, which I stated before. So then we got more specifically into the dural venous sinus CSF drainage. This mechanism is operating when there is an increase in intracranial pressure and not under normal conditions. So this is why this one's not as important as the lymphatic CSF drainage. So the cerebrospinal fluid here enters the dural venous sinuses through the arachnoid villi, or the arachnoid granulations. The arachnoid villi poke through the dural wall of the sinus, and it's thought to be a one-way bulk flow of CSF across the villi into the sinus. But this mechanism can only happen or operate under conditions of increased intracranial pressure. So what happens if we have the destruction of normal CSF flow and drainage? So I told you how the CSF, so on the right, the picture on the right, I've already talked about this, is how the cerebral spinal fluid flows. But however, hydrocephalus is when, if you see this puppy on the right, there's too much cerebral spinal fluid in the brain or the skull. That is what it's called, hydrocephalus. The third ventricle is expanded and the lateral ventricle is also expanded. So what if we block the cerebral aqueduct? This is why this hydrocephalus occurs, because then all the cerebral spinal fluid backs into the third and lateral ventricles, and the fluid just keeps building up. This is referred to as a non-communicating hydrocephalus, which causes compression of the surrounding brain tissue and an increase in intracranial pressure. There's also communicating hydrocephalus, which is when there's a blockage of the cerebral spinal fluid, absorption and drainage from the subarachnoid space, which therefore increases intracranial pressure. So how does CSF help with brain waste disposal? Sorry, let me get to my slides real quick. The CSF is continually produced in the brain at a constant rate. It turns over several times a day. There is a liberal exchange of solutes between the extracellular fluid and the cerebral spinal fluid. There are perivascular spaces around the blood vessels, arteries in the brain that are filled with the CSF fluid. This in turn helps to wash across the cells in the brain. It is then carrying away waste products and carried into the perivascular space in the veins. So it's that perivascular flow of the cerebral spinal fluid that washes away across brain tissue to carry away interstitial metabolic waste. So then we got into how does the brain get oxygen and nutrients to produce energy? The brain gets these from the blood supply. The brain uses actually 20% of the blood supply. So what are the major arterial sources bringing blood to the brain? Well, the vertebral arteries and the carotid arteries. The vertebral arteries enter the spinal column of the first segment. The carotid arteries enter the skull from the branches of the common carotid. These two supply a network of vessels on the ventral surface which distribute throughout the brain. So we have the artery, basilar artery, which runs down the midline of the medulla and pons, which gives off little branches that are running into the brain tissue of the medulla and pons, which therefore supply the blood. So there are two cerebellar arteries, which are rostral and caudal. The caudal one comes off the basilar artery, which supplies the caudal portion of the cerebellum. The rostral cerebellar artery supplies the rostral portion of the cerebellum with blood. But then there are also three cerebral arteries, the middle, rostral, and caudal. The middle one supplying most of the lateral portion of the hemisphere of the cerebrum. Then there's the rostral, which branches off the internal carotid, which supplies the rostral medial portion of the hemisphere. It also supplies a small portion of the rostral lateral portion of the hemisphere. And then there's the caudal cerebral artery, which branches off the caudal communicating artery. It supplies the caudal medial region of the hemisphere. and it also supplies a small portion of the caudal lateral portion of the hemisphere. So what are some ways the brain maintains the consistency of its blood supply? Well, the cerebral arteriole can rapidly change in diameter in response to systemic blood pressure. If there's a decrease, they will get wider, so blood flow to the brain is constant. And if increased, the arterioles will constrict their diameter. The blood arterioles can sense change in carbon dioxide and oxygen. However, they're more sensitive to the change in CO2 than they are in oxygen. They can also locally change their diameter, which changes in neural activity. So more activity means a wider diameter. So remember, cerebral arteriole changes diameter. So there's always a consistent amount of blood supply. So how does the brain prevent access of potentially harmful disruptive chemicals? The capillaries have liberal exchange. The brain capillaries are more restricted and selected. There are tight junctions between the endothelial cells, so they don't have open clefts. They don't have the little holes for smaller materials to pass through. There is very limited non-specific transport. The main things that can pass are the lipid-soluble substances without assistance. Other things require specific carriers to carry other molecules across. They are usually energy dependent. And there's an increase in mitochondria in the walls of the cerebral capillaries. The blood-brain barrier keeps unwanted material from blood to the brain. However, the blood-brain barrier can be a mixed blessing. It can allow addictive substance through without having an aid. And therapeutic drugs have a hard time sometimes getting through because of this barrier. So, yeah. So I think that was all for neurobio. And I think nope. Sorry, we still have like 5 slides. So then we got into the overview. So this is neurobio 5. Then we got into the overview of structure, function, and pharmacology of the autonomic nervous system. So what is the autonomic nervous system? Well, let's see. So the autonomic has to do with the cardiac muscles, the glands, and the smooth muscles, and its main function is to keep homeostasis. The somatic nervous system has to do with skeletal muscles, and it is external. So what are the major divisions of the autonomic nervous system and their functions? So we have the sympathetic and parasympathetic. The sympathetic being it prepares the internal environment for the emergency energy expenditure for emergency situations. It can increase respiration. It can shut down things. Basically, it's a fight or flight response system. So it revs up, basically, the nervous system, whereas the parasympathetic brings things back to the baseline level. So bringing things back to basically homeostasis for restoration and conservation of energy in non-stressful times. So this is known as the rest and digest system. So then we have an enteric division, which is also known as the second brain. It can be considered a third division of the autonomic nervous system. It is part of the GI tract wall and is a self-contained system of sensory and motor neurons that can independently maintain the GI function. However, the enteric division function can be influenced by the CNS, the central nervous system, by way of the sympathetic and parasympathetic divisions. So what are some differences between the somatic and automatic motor systems? The somatic is controlling skeletal muscle, like I said. It's usually conscious of these actions. Actions at the peripheral target is always excitable. There's only one synapse between the central nervous system and the peripheral target. The autonomic has to do with controlling the cardiac smooth muscle and glands, like I said. It's unconscious to these actions. Actions at the targets are either excitable or inhibitory. There are two synapses between the central nervous system and the peripheral target. So I think that's all we got to. We didn't get very far into this lecture, so he'll be lecturing us again tomorrow. So the last lecture we got into is, I believe, the heart. So the guy who teaches this microanatomy lab, his wife actually teaches cardiac. So I thought that was really cool. So our main objectives now are to understand the physiological purpose of the cardiovascular system. We have to define the components of the CV system. So cardiovascular, I'm going to use CV, and discuss the unique characteristics of the CV system that support its purpose. So basically, we talked about in class purposes of the CV system. Is to transport oxygen to the tissues. It also delivers nutrients. It works with the endocrine system, so hormones are circulated. It has to do with temperature regulation, circulation of the immune system. So it has to do with inflammation, so it can increase blood flow to the area. It can redirect blood flow. It has clotting factors and platelets, and it also gets rid of waste. Waste products such as CO2, so it can keep a pH balance. So basically, it's the circulation of blood, delivery of what the cell, tissue, body needs, and the removal of what the cell, body, and tissue doesn't need. So what does this body need exactly? I'm going to show you pictures. So if a running horse. So it's basically an exercising horse. So it definitely needs temperature regulation. It needs cooling mechanisms. So it moves where the blood is located. It moves more superficial and opens up. So that's a cooling mechanism. So this horse's muscular activity is creating a lot of heat. Therefore, his muscles have increased energy requirements and waste production. So the answer to what does this body need is this body needs regulate blood flow to the areas of need, increase blood flow to high metabolic demand tissues like the skeletal muscles to deliver oxygen and remove CO2 as well as other products, regulate body temperature through convective cooling, the dilation of the epidermal vessels, and it preserves blood flow to essential organs such as the brain, heart, and kidneys. So then we got into another picture of what does this body need. And here's a dog. Here's our picture. And the answer to this is the body needs a delivery of needed products, platelets to help stop the bleeding, regulated blood flow to reduce blood flow to the area of injury, so vasoconstriction, and an increased blood flow to the essential organs such as the brain, heart, and kidneys. So then we got into the components of the cardiovascular system. So we have the arteries, which deliver blood from the heart to the tissues. Then we have the capillaries, where exchange occurs. It is the business end of things, so to speak. It's really essential, even though we rarely talk about them. And then we have the veins, which return the blood to the heart. And then we have the heart itself, which is the central pump that moves blood forward. So So here's everything that I just talked about. So the deoxygenated blood returns to the right heart pulmonary circulation to take up oxygen and drop off carbon dioxide at the pulmonary capillaries. This is known as pulmonary circulation. And then oxygenated blood leaves the left heart to go to the systemic circulation to deliver oxygen to tissues and take up carbon dioxide at the tissue capillary bed. This is systemic circulation. So then we got asked a question, which of the following is true? All arteries carry oxygenated blood, or all arteries carry blood away from the heart? So we learned it is all arteries carry blood away from the heart. So again, we got into more components of the cardiovascular system. We have the vena cava, which is venous return to the heart, deoxygenated blood, cranial and caudal vena cava. And then we have a 4-chamber heart. We have the atria, which hold blood until they go through the ventricle. It has a minimal pumping function, but it's a conduit and booster filling function. And then we have the ventricles, which have a thicker wall than the atria. They have the pumping function to the pulmonary and systemic circulations. And then we have the pulmonary arteries and the aorta. The pulmonary artery and aorta, arterial delivery to circulations. And which one has oxygenated blood? That would be the aorta. So the venous return, the deoxygenated blood, which is in the blue here, it returns to the right heart through the cranial superior to the cranial vena cava and the caudal vena cava. Blood enters the right atrium when the ventricles are filling, which is known as diastole. Blood flows from the right atrium to the right ventricle through the tricuspid valve, which is also known as the right AV valve. Which is the AV means the atrioventricle valve. So when the ventricles are pumping systole, blood flows from the right ventricle through the pulmonic valve, which is also known as the semilunar valve, to the main pulmonary artery, through the pulmonary capillaries to obtain oxygen and remove CO2. So now that the blood is oxygenated, where it turns red, this oxygenated blood returns to the left atrium through the pulmonary veins. Note that the venous blood is usually deoxygenated, so pulmonary veins are unique in this respect. But once in the left atrium, when ventricular filling, again, diastole occurs, blood will travel through the open mitral valve, the left AV valve, into the left ventricle. During ventricular pumping, oxygenated blood is pumped from the left ventricle through the aortic valve, also known as the semilunar valve, into the systemic circulation. So again, definition: systole is a marker of time pumping blood into the system, and it always refers to the ventricles. Diastole is when ventricles are relaxing and allowing ventricle to fill with blood. So does systole pumping of the atria and ventricles occur at the same time or sequentially? So the answer is sequentially, because one has to fill with blood, which would be the atria, and then it will pump into the ventricles, and then take that blood and pump it forward. The atria will contract first, and then ventricles will then contract. So then we got into the components of the CV system: a central muscular pump with unidirectional valves supporting forward blood flow. Valves can only open in one direction. The heart has a base, which is the atria and great vessels, and it has an apex, which is the left ventricle. the left ventricle hits the wall of her chest, and that's why we can feel it. The right ventricle is near the apex, but is not defined as the apex. So then we have the AV valves, the atrioventricular valves. They separate the atria from the ventricles. They have a tricuspid, which is the right AV valve. They have the mitral, which is the left AV valve. And they open for ventricular filling diastole. And the AV valves have to be open. And then we have the semilunar valves, which separate the ventricle from the great vessel. They have the pulmonic and aortic. They open during ventricular pumping, systole, when ventricles are pumping to deliver blood to the pulmonary and systemic circulations. So which valves open and close during systole and diastole? So during systole, the AV valves are closed and the semilunar valves are open. During diastole, the semilunar valves are closed and the AV valves are open. This is so blood can fill the ventricles. So what tells the heart to beat? Because hearts can still beat even outside of our chest. So that's why we do heart transplants. They can still be alive and still beating without being connected to us. So that is known as the sinoatrial node, which is the heart's pacemaker, also known as the SA node, in the right ventricular area. It's specialized cells that depolarize spontaneously. A depolarization is what starts contraction. The Purkinje fibers send down this depolarization. So again, the spontaneous depolarization at the sinus node initiates events that result in mechanical pumping. So then we got into myocytes. Which are components of the CV system. We have specialized cardiac myocytes of the cardiac conduction system. We have which includes the sinus node, the conduction systems. But then we have working cardiac muscle myocytes, which are responsible for muscle contraction and pumping of blood, and they make up most of the atrium ventricles. So again, the sinus node depolarization initiates the cardiac cycle. So the events of each heartbeat. The atrial depolarization and contraction enhances ventricular filling diastole. The AV node is a pause between the atrial depolarization and ventricular depolarization to optimize ventricular filling before ventricular pumping starts. Ventricular depolarization and contraction results in ventricular pumping, systole. So we have coordinated contraction, systole pumping, and relaxation, which is dystole and filling of cardiac chambers, results in the forward blood flow with each heartbeat. The conduction system helps to coordinate this series of events. So basically that was cardiac 1. I don't even think we got into cardiac 2 today. I don't think we did at all. So that was it for today. I'm pretty sure. So we learned some really interesting things today. My day is not over. I had to do integrative stuff, and I still have to exercise, and still have to eat, and still have to do study. Yeah. But I hope you guys all had a good day today. And I will I will talk to you tomorrow. But just to show you, my dogs are still alive. Here's Lillian. There's Sky over there. Mermai's in her pineapple over there. But yeah, we're all still alive over here. But yeah, I love you guys, and thank you guys for watching.